Hi everyone, thanks for checking out this video. So this is going to be a video about how to write a Python program to convert RNA-seq raw count data into what's called TPM data. That stands for transcripts per million. And that's a common normalization technique that's used on a lot of RNA-seq data sets. And so before I get started with showing you guys the Python code, I just first want to recommend a video to check out that explains like why TPM is a good normalization technique and why it's kind of like preferable to other normalization techniques for RNA-seq. So I was thinking if I should try to explain this myself, but then I just thought that this video explains it better than I can. This is from the StatQuest channel, by the way. Really good channel with lots of like statistics and bioinformatics videos. So I'm going to recommend, if you guys haven't seen it, if you check out this video for like an explanation of why this is a good normalization technique and sort of like a good intuitive like visual explanation. But so my video is going to be more about like how to implement this normalization in Python. So we're going to be writing a Python program to convert raw RNA-seq counts into the TPM dataset. So before we get started on the code, I'll show you guys what kind of data you need to do this. So you really need two components to the data. So you need the actual raw count data set. So I'll show you guys what that looks like. So this here is it's just like a fake data set I made for these genes. I'm just calling G1, G2, all the way up to G100. So each column corresponds to the counts for some gene, and then the rows correspond to different samples. And if you have an RNA-seq data set, one clue that it might be just like a raw count, unnormalized data set is that the data is in integers. So since we can see all these are integers, that's kind of a clue that these are just like the raw counts that haven't been normalized. So yeah, just to reiterate, we have a column for each gene. I've just called them like G1, G2, up to 100. And then the rows are different samples that we have. And I think I put in like 500 samples here. Yeah. Okay, so that's the first thing we need. So this is just kind of our raw count RNA-seq data set. The second thing we need is we need to know the lengths of all of the genes in bases. So I have this thing here, the lengths.csv. So this is just saying for each gene, how long is it in bases? So this is literally talking about just like how many nucleotides make up this gene. And we need this because this is one of the things we're going to be controlling for. So we need to control for the actual length of the gene. And we also need to control for sequencing depth. But yeah, sequencing depth we can kind of calculate. But for the length of the gene, we need to actually have some kind of file that tells us this information. Okay, so these are the two things we need, and then so we're going to import kind of some of the usual libraries we're going to be working with. So we're going to import NumPy, SNP, CSV. We actually don't need this. I just had that when I was debugging a bit, and then I'm going to use pandas to save the new data at the end. So yeah, import pandas SPD. Okay, so just to get started, first thing we're going to do is just read in our data. I'm going to be a bit lazy here like I usually do. Just going to copy and paste a little bit of this code just for the reading in the CSVs. So this is reading in the length CSV, and so very importantly, we're going to save this. We're going to save this data as a dictionary rather than a list, because I know in most of my videos I usually save it as a list. But here it's going to be faster to like call this data when we need it if we save it in a dictionary, and you guys are going to see in a bit why that when that comes into play. But yeah, so we define an empty dictionary, the length dictionary, and then we're just going to open our length.csv file as just CSV file. We're going to define our CSV reader. Delimiter is a comma because it's a CSV. If it was like a TSV, then this would be a tab. And then we don't have any header in this file. But if you did have a header, you could just say something like next parentheses reader to skip over the header, but there's no header here, so I don't I left that out. And then we're going to iterate through each row in this CSV. And for each row, we're going to make a dictionary entry to have the first element of the row, which is the name of the gene. We're going to have that correspond to the second element of the row, which is the length. But we need to make sure we cast this length into a float, otherwise it'll just be right in as a string. So we cast it to a float here. And if you guys don't understand like the point of like a dictionary data structure, it's basically just for when you want to have, when you're reading in data where like one thing corresponds to something else. So here we want the gene name to correspond to a length. So we're just saying, this is the syntax for that here. We're just saying like, okay, the length dictionary entry for this gene name is going to correspond to this gene length and casting that to a float. So okay, hope that makes sense to you guys so far. So that'll read in all of our gene lengths. So the next thing to do is actually read in our data. So just remind you guys, like we have here, it's just the first row is just the names of all the genes, and then every row after that is our actual data, our raw counts for these genes for each sample. Okay, so I'm gonna again be a little bit lazy and copy and paste some code here just for reading in the CSVs. So here we're just going to define an empty list called data, and then yeah, just open up the CSV like we did before, define our reader like we did before, and then for each row we're going to append the row to the data. And so far at this point it has read in the header, and it's also reading in everything as strings. So we need to also correct for that. So we're going to say genes is data, the first row of this data list we just filled up. So that's just going to save genes as the names of all of our genes here. And then we're going to say data equals data starting with the second row onward. So that's just going to resave our data variable as being everything from this second row down, because that's where the actual data is. So we basically just save the first row in this genes variable, and the second row downward, we're keeping that as our data variable. And then remember, this is still in strings at this point, so we need to cast everything to a float. And what I'm actually going to do is I'm actually going to cast it to a NumPy array just to make it easier to work with. And then I'm going to cast that entire NumPy array to the float data type. So I'm just saying, okay, data equals np.array of data, and then as-type float. So that'll just cast it to a NumPy array, and then cast everything in that NumPy array to the float data type. Okay, so at this point we've read in our data. Maybe at this point I'll actually just print it out and make sure it's okay. Yeah. Yep. So it looks like you're right in. Okay. Just to remind you guys too, we could have cast this to the int data type, because remember all of these are integers, but I'm casting it to be floats because I just think it's a if you're not worried about memory, and in this case I'm not really worried about like running out of memory or something, I think it actually is good to just play it safe and just use the float data type instead of the int data type. If you're worried about memory, then yeah, you want to leave them as ints because it won't take up as much space. But if you're not too concerned about memory, I think it's a good habit to just be working with floats in case you do have something that that isn't an integer. Yeah, but okay, so moving on. So now we've read in our data, so the next step is to actually do the conversion to TPM. So I'm going to copy and paste this comment I left, actually, just a comment just describing the steps. And again, I recommend checking out that video if you guys aren't familiar with like the concept of TPM. But I'll try to explain a little bit here too, even though I think the video explains it, the StatQuest video, I mean, it explains it better than I probably can, but I'll try to explain a bit too. So when we have our raw counts, the first thing we need to do, there's really two steps. The first step is normalizing for the gene length, which is going to be in kilobases, which is thousands of bases. So that's going to give us a measurement called reads per kilobase, or RPK, if you ever hear about that, it's just reads per kilobase. That's what we get when we normalize for the gene length for all of our counts. And then once we have that, the next thing we're going to do is for each sample, we're going to add up the total RPK, and then also divide that by 1,000,000 to get this transcripts per million scaling factor. And that's going to give us a measurement that we can divide by to normalize for sequencing depth for all of the samples. But yeah, you guys this is going to make more sense, I think, when we actually implement it in the code. But yeah, it's just 2 steps here. So the first step is normalizing for gene length, and the second step is normalizing for sequencing depth. So again, if you guys are confused at this point, I mean, first of all, I recommend checking out the StatQuest video, and secondly, I think this will make more sense when we actually implement it in Python, so you can actually see what's happening. Okay, so let's just start with step 1 here. So the first thing we're going to do here is we're going to define an empty NumPy array of zeros that we're going to use to fill up our RPK data. So we're just going to call this RPK, and it's going to be an array of zeros with this shape. The shape is going to be the same shape as our raw data, so it's going to be data shape 0 by data shape 1. So we want this RPK array to be the same shape as our raw data, except full of zeros. And we're going to fill this up with the RPK measurements. So we're basically going to have so we're basically just going to use for loops to do this. We're going to use for loops to just iterate through all of the rows, and then a nested for loop to go through all of the columns. And then for each data point we have, we're going to divide by the length of the gene of that data point, and that's going to give us our RPK measurement. And we're just going to fill up this array of zeros with the actual RPK data points. Okay, so we just need 2 for loops here. So for I in range data shape 0, that's going to be going through all the columns. Second for loop, it's going to be nested in here. For j in range data shape 1. Quick note here, it's actually I'm doing this for simplicity. When I'm keeping things simple, I kind of like to just have the first for loop be going through the rows and then the nested for loop be going through the columns. In this case, it's actually going to be like slightly faster if you do the opposite. If you have the outer for loop be going through the columns and the inner one be going through the rows, because then you just won't have to make as many calls to the length dictionary. But for now, just for simplicity, even though it's going to be a little bit slower, I'm not super worried about the speed here. So just for simplicity, I'm going to be doing it like slightly more slowly, just in a way that makes sense, just going through the rows and then columns rather than columns and rows. But yeah, so now for each of these data points, like we're going to be going through, this is going to be giving us access to each of the data points by index. And then for each one, we need to know the length of the gene that we're talking about here. So we're going to say gene length equals length dictionary, and then we're just going to make a call to the genes. We want the jth gene, because this inner for loop is going through is going to give us just the index of whichever gene we're talking about. Remember the genes are columns here. We're getting a value of j for each one of these columns, and then each column corresponds to a gene. So if we just make a call to the genes list here, and then see what the dictionary value is for that gene name, that's going to give us the length of the gene. And the reason I said it was going to be a little bit slower to do it this way is that when we're doing it like iterating through the rows and then the columns, we need to make a call to the dictionary for every single data point. Whereas if we did it in reverse, then we could do something like putting this up here, so we'd only have to we wouldn't have to make all these unnecessary calls to the dictionary. We just like for each column we would just find the gene length and then go through all of the rows and then apply it. So yeah, I'm doing it slightly more slowly here, just because I mean in this like test example I'm not super worried about the speed, so I'm just kind of like doing things simply to be able to teach it and hopefully have you guys understand it. But if you guys were super worried about speed, you could actually reverse these for loops and have the outer one be the columns, and then you wouldn't have to make like unnecessary calls to the dictionary. But yeah, just like I said, for now I'm just kind of keeping it simple, not super worried about speed, so it's okay to make these redundant calls. Okay, so this is giving us our gene length. So what we need to do next is this is the length in bases. We need to get it to kilobases. So I'm going to say gene length in kilobases. So basically, this is just going to be the gene length divided by 1,000. This is just something that makes it easier to talk about because a lot of the genes are like very long. They can be like hundreds or thousands of bases long. We just want to get it to something like easier to work with. So we're dividing by 1,000 to give us kilobases. That's also just kind of the standard for RPK. Yeah, we're not talking about reads per base. We're talking about reads per kilobase. So we need to divide by 1,000 to get it in kilobases. Okay, and then after we do that, so we're going to say that our RPK array at the index ij is going to be equal to our data point at the index ij. So remember that's our number of like raw counts, the unnormalized data. All we're going to do for this step is just divide by the gene length in kilobases. That's all we're going to do. So super easy step for this first step. Just filling up this RPK array. All we need is the length, and we divide the length by 1,000 to get it in kilobases. And then we divide our raw data by this gene length in kilobases and store that value in our RPK array. So yeah, for the first step, that's really all we need. Pretty easy so far, if you ask me. Nothing too complicated. As long as you have all the data and then you have all the length and stuff, it's pretty easy to implement. Okay, step 2. Hopefully this will be just as easy. So the first thing we're going to do is now we have an array for our raw data and we have an array for our RPK data. We just need one more array of zeros that is going to store our actual TPM data. So again, it should be the same shape as the other two arrays. It's going to be, yeah, like a row for every sample and a column for every gene, just keeping the same shape as before. But I'm just going to call this our TPM array because we're going to be storing our actual TPM values in here. Okay, and we're basically going to do kind of the same thing. Just we're going to do this like this 2 for loop, one of them nested kind of thing. So we're going to say for I in range RPK shape 0. You could say data shape 0 here because remember it's the same shape. But since we're going to be working with the RPK array, I think it kind of is helps us keep track of things to use RPK here. But remember, it's the same shape for all of them. Could have said TPM here, same shape for all these arrays. Could have said any of them. But yeah, just giving it RPK here. And then yeah, it's going to be spacing it out because we're gonna actually put something in this space here, but we're going to also go through the columns for J in range RPK shape 1. So this is going to be our nested for loop here. But for each sample, remember the I's are going through the rows, which are the samples. For each sample, we're going to calculate the total RPK. So we're going to say total RPK equals np.sum. And we're just going to sum up the RPK for this row. Basically saying for this sample, we want to get the total sequencing depth for this sample that we're talking about. So we're just summing up this entire row in our RPK array corresponds to the RPK for this sample. Okay, so that's pretty straightforward so far. Just our total RPK, and then we're gonna then scale it by 1,000,000 because we need our final answer to be like scaled like per million. So I'm just gonna say, because I mean that's just part of the name, like TPM transcripts per million. There needs to be a per million at some point in here, right? So I'm just gonna say scaled RPK equals total RPK divided by 1,000,000. Yeah, okay. And here we can take advantage of like not having to make all these like redundant measurements, because here it's like we can do these. We don't have to do these for every single data point. We can do them in the outer for loop because we're going through all of our samples, and for each sample, these are going to be the same like for every column. So we can leave these in the outer for loop to make it a little bit faster. But okay, once we have those, then we're just going to go through all of the columns here. So remember, J is iterating through through the columns, and each column corresponds to a gene. So then we're just going to say TPM, TPM at the IJ index equals RPK at the IJ index divided by scaled RPK. Sorry, maybe I should just to keep the naming make sense, maybe I'll actually say scaled total RPK. Change that for both of these actually, just so we remember it's a scaled total that we're talking about here. Okay, so this gives us our TPM measurement, but if you guys are thinking ahead here, you might realize that for some data points this might actually give us an error. So this could give us an error if any sample has 0 for their like total RPK. If any of these is a 0, we're going to be getting a like divide by 0 error. And so sometimes you just get like, especially in like single cell RNA-seq or something, you might get an entire sample that just didn't pick up anything. So for some of the samples, you may actually be getting like a 0 for one of these. I hope you're not getting like too many of those, because it's kind of not good if you're just not getting any any sequences. But in single cell RNA-seq, you may be getting a couple of these like dud samples that don't have any reads or anything. I always just throw in an if statement here. I say if scaled total RPK doesn't equal 0. If it doesn't equal 0, I say to do this to get this TPM measurement, and then if it does equal 0, I just say TPM IJ equals 0. Or even just to have this make even more sense, I would just say that it equals the RPK, which is going to be 0. But just to have it make more sense, maybe I'll write it like that. But okay. But yeah, this should be it. As long as I didn't make a mistake anywhere, this should be all we need to do. So just the last thing is just to save this data to a CSV. So we just want to like save it in kind of the same format as our starting data. We just want like a header with all the gene names, and then we just want all of our TPM measurements for each sample. So for this, I'm actually going to use pandas, and I know in my videos I don't use pandas that much because, to be honest, I don't like pandas that much. I don't find it to be like super helpful for a lot of things. But for this, for saving a CSV, if you already know like a nice format you want it in, I actually do find pandas to be helpful for this, like saving a CSV. Okay, so we're going to say pandas.dataframe tpm to CSV. Just call it tpm.csv. Header equals genes. We just want the header to be all the names of the genes. And I'm going to say index equals false here. We don't want an index for it. Okay, so I think as long as I didn't make a mistake, this should be all we need to do. So I'm just going to run it and hopefully it works. Okay, looks like it worked okay, but let's just check to make sure. Just check our output CSV here. And yes, so it's I know it looks like super messy, but actually this is kind of what it's supposed to look like when it's in this normalized format. As you guys can see, like we don't have these nice looking like integer counts anymore. We have actual like normalized floats. So I know it doesn't look as nice, but this is actually what it's supposed to look like. So yeah, looks like it worked okay. Just to quickly review everything we did. So we just read in our data. To do this, you need some kind of raw count data set, and you also need to know the lengths of all the genes you're working with. Yeah, sometimes if you're getting data from like a collaborator or something, or downloading it from like a paper or something, this may be tough to get a hold of, or you might have to kind of like look around for it. But yeah, getting the length data, you need it to do the TPM normalization. But yeah, so we just read in our data, then 2 steps for the TPM normalization. So the first step is just normalizing for gene length in kilobases. That gives us our RPK measurement. And then the second step is normalizing for the sequencing depth. So basically for that, we just divide by the total RPK, but we have to remember to, since we're talking about transcripts per million, we want to also just scale our total RPK by dividing it by 1,000,000. But yeah, then we just go through and divide all the data points by that. But yeah, that's really all there is to it. Then after this, you have this nice like normalized data that's like easier and makes more sense to work with. Again, if you guys don't understand why we're doing the TPM normalization and like why this is important to do, then I recommend checking out this StatQuest video. Great video, good like intuitive visual explanation of why this is done. But yeah, if you guys just need to know the Python code to do it, I hope this video has been helpful. Just like writing the Python code to do this normalization for our RNA-seq data. Okay, guys, thanks for watching. Hope this video was helpful to some people. If you have any questions, just let me know in the comments, and I'll try to answer them. Also, like usual, all my code will be on my GitHub if you want to download the code and like practice it for yourself. But okay, guys, so thanks for watching, and see you next time.